who have previously been joining our Toxics Pod conversations, and some new folks too. So I want to welcome the new folks and just provide a little bit of a context for you so you understand some of the acronyms and things that we'll be talking about this morning. So the Toxics Pod is a work group that the Strategic Initiative manages to help inform our work for Puget Sound recovery. We want to take a broad national perspective, but really kind of have a narrow focus on Puget Sound recovery. So we're looking to bring in new ideas and learn from people both inside and outside of Puget Sound, like we have some of our guest speakers from outside of the area this morning. We're also wanting to connect communities of practice, whether that's public health, ecotoxicology, stormwater, policy, management, all of those things. We're looking to be a place where we can connect on that and really kind of make this a science to policy interface where we can learn from the science to inform policy and help scientists and researchers learn about the types of policy questions we have and needs that'll help them direct science to accelerate recovery overall. Today, well, really, this focus that we have today and that we'll be continuing with here for the next couple of well, the next year at least, is to focus on integrating human health and toxics in aquatic life. Really, it's come down to breaking down silos and bridging gaps, communicating and learning from each other so that we can develop holistic approaches to solve complex problems in Puget Sound. Those of you who are new to this group, you'll hear this acronym Toxics in Aquatic Life, or TL, or Toxics in Fish. These are this is an implementation strategy that was built a couple of years ago and is meant to inform the Puget Sound recovery efforts and our investment of EPA geographic funds. The graphic on the left side of your screen is the Puget Sound vital signs diagram. You'll see where the star is the toxics in aquatic life. Our Stormwater Strategic Initiative covers this implementation strategy as well as the implementation strategies for marine water and fresh water there in healthy water quality. But it's important to note that these strategies do affect other parts of our vital signs, and that we've been working on and connecting the thriving species and food web there in the bottom yellow box. That's kind of our main goal, and we have indicators that you'll see on the upper right for contaminants in salmon, mussels, sole, and herring, representing different niches of the marine aquatic environment. So we have those indicators, and we have strategies that focus on helping people reduce their toxic exposure. And we're trying to figure out how to elevate and include more of those healthy human population goals and objectives in the work that we're doing. And also, recognizing that it's not just human health, but also human well-being and economic vitality that's improved by these actions. So know that when we're talking about toxics and fish implementation strategies, this is going to refer to a document. There's links in your meeting packet if you want to dig into that, if this is fairly new to you. But feel free to ask questions along the way today and get clarification if you need it, if this is new to you. I'm going to hand it over to Heidi here for some integration advice.
Okay, here we are this morning to do some silo busting. So every single one of us has been trained in a particular way. We have our communities of practice. We have schooling, education. We belong to associations. We go to conferences, and that sort of forms our entire view about the world. And I think the general proposition is that silos, although functional and can be convenient from an organizational standpoint, have a lot of traps associated with them. This is a great article. If you have a chance to pick it up, there is a link in the notes section of the PowerPoint, which we will share. Next slide, please. So understanding the dynamics of adult group learning, change, networks effect, cognition, bias, and it really starts with humans, their behavior, and how they learn and change over time, trust and self-efficacy, and having the skills and believing that when you do X, Y will result. And on the bottom of the page, you'll see an article on factors that encourage One Health collaborations, and the throughput strategies that make this possible include obviously leadership, whether you're really interested in working with people from other fields, and broader network structure, and importantly, the ability to share data and remove some of the operational boundaries between sharing that data. Next slide, please. So last summer, we brought together a group of practitioners, and a few of you on the call were there. We wanted to look at how human health, toxics, 6-PPD, turf, and solid waste might all view the same issues in very different ways. And so Dustin very nicely put together this AI generated image, and we gave people an opportunity on a mural board to look at what they were seeing when they saw this image. So that was a very interesting exercise. And as you can see here, there were very different things that people actually saw. Next slide, please. And so this is just a snip from our mural board. And we asked people about operational boundaries, risk tolerance, actually, and what was getting in the way, but also what they were optimistic about. And so, you know, as we find out that 6-PPDQ, the tire wear particle, Eleanor Fanning, who's on the call, is doing some human health research there, but there's also very strong connections to turf, which is going to be the next topic of discussion with the Safer Products in Washington law. So we're very excited about that. Next slide, please. And then this is from pedagogy, from, you know, learning for younger students, but I really liked this benefits. You know, you can consider all of us students because we're students of life, because we continue to learn every day in our fields. And I really like this a lot. So basically we become more effective, more holistic. Our decisions will be better, and they'll also be more, I think, politically palatable, to the extent that we're looking at not only effects on ecosystems and animals and plants, but also on human health and what the economic ramifications are. And so on the right, you'll just see a couple of collaborative entry points. This would deserve a 3-hour meeting by itself, but I'll leave it at that. And I think that's it for me, Dustin.
All right. So after that meeting last July or June, last summer, we had a workshop in December. We had great turnout and a lot of input from everybody. And that was really designed to help figure out what is our vision statement for integrating human health or a One Health approach with toxics and aquatic life implementation. And came up with these kind of main areas of focus. So first off is really developing a human health implementation strategy within the Puget Sound recovery system. And that can take a lot of different approaches. It could look something like this Toxics and Aquatic Life IS. It could be something much simpler. It could be new strategies within existing implementation strategies as well. So, but the idea of having a platform or a landing space for these human health issues was identified as important. Also using a transdisciplinary problem-solving approach. And that's part of our silo busting activities. Our investments in human health research and toxics reduction, and we'll talk a little bit about that later this morning. community engagement, science communication, all of these things. There's a draft vision statement that I included a link to in the meeting packet. And this is just a very draft right now. So I'm welcome to receive your feedback on that by June 30, just so I have kind of a deadline to work with and update the vision statement. You can go to the meeting packet, the link in your meeting packet, and make track changes right on the document, or read through it and send me your suggestions for changes. Comments really are better than track changes in that it makes the document easier to read. But really looking for feedback on, is it clear? Have we missed anything important? And is it scaled appropriately for toxics and aquatic life implementation? Knowing that some of the things in there are beyond the scope of toxics and aquatic life, but are also important for this integration practice. So this is my last slide, and then we'll get over to our speakers here. Just want to lay out the next steps for this is that this Toxics Pod is going to continue learning from examples inside and outside of the Puget Sound region. Going to be setting up some future meetings here for discussing how we can better collaborate and share information, and continue just to foster a transdisciplinary network of practitioners. I see your comment, Kathleen. Yes, we'll make sure you get that meeting packet in a link in the chat here in a moment. And then beyond the Toxics Pod, the Stormwater Strategic Initiative team is going to be working within the Puget Sound Recovery Network to develop a platform for human health and toxics reduction. Not sure what that's going to look like yet. As like I said, it could be a lot of different things, and there's a lot of moving pieces that are moving right now, and so it's just going to be a matter of kind of seeing how everything shakes out and how this will look going forward. But really, it's going to mean adapting or updating our toxics and aquatic life implementation strategies to better include human health objectives. And with that, I'd like to turn it over to Heidi, who will introduce our speakers.
Yes, thank you so much, Dustin. I now want to introduce Dr. Catherine Karr, who runs the University of Washington's Pediatric Environmental Health Specialty Unit, which is our regional PEHSU here in the Pacific Northwest. We're thrilled to have you, Catherine, and we appreciate the time that you're taking. We know that you're very busy, and and things are very interesting now. Please join us, and you can share.
Great, thank you. Trying to do the share screen now. It says you guys can see my screen. It's interesting because I can't see it. It's telling me. I'm not sure what that's about.
Let us know. Oh, there you go. Okay.
Okay, now I can. It's a little slow this morning, warming up, I guess. Well, hi, everybody. Thank you so much for inviting me to participate. And I think this is a really appropriate talk for the breaking down silos concept, because I suspect most people on this call have not heard of the pediatric environmental health specialty units. Sometimes I think we're just a little bit under the radar, and we really welcome the opportunity to get the word about, who we are, what we do, and think about partnerships and shared goals and work activities. So I am at the University of Washington. I'm a pediatrician and environmental health researcher. And one of my roles here at the university is to lead something we call the Northwest Pediatric Environmental Health Specialty Unit. And if you haven't heard of it, this is part of a national network, and it comprises individuals across the country who have special expertise in reproductive and child health related to environmental exposures. And if you go to our national website, PEHSU.net, you'll see, these words flash on the screen. They are based at academic medical centers, so there's one in every federal region. And we work collaboratively to improve health systems and to make communities safer for kids and families. And our activities are in the realm of providing education and outreach, training for health professionals, guidance for health professionals, and we actually do consultations or provide specialty referrals for patients. Our funding source comes from the federal agencies. The bulk of our funding comes for the Centers for Disease Control through their Agency for Toxic Substances and Disease Registry Program. And it's all the acronyms, like a lot there, but ATSDR, and then some funding from US EPA as well. And we're centrally coordinated by an organization called Public Health Institute that kind of helps all these 10 regional PEHSUs be connected. We've been around a long time. The University of Washington was one of the first PEHSUs. There were only a couple in the beginning, but now, as I mentioned, they're in every federal region, and that's the national website there. So you can see, you know, many of you are probably familiar, or if not, these are the sort of federal regions. So here in Region 10, is where Washington State is housed, along with Alaska, Idaho, and Oregon. So we serve all 4 states. And we also have our own local website, Northwest PEHSU. So I welcome you after to get to know us a little better, to take a look at both the national website and our local version. And if you want to contact us later, you have a consultation or a query, we use PEHSU@uw.edu. And we also have a phone number there as well. So to give you an idea of the specialty and expertise here at in our Northwest PEHSU, in addition to me, and I'm, you know, my specialty areas of expertise are around air pollution and wildfire smoke, pesticides, lead exposure. I do get involved kind of in everything, but those are areas that I have, you know, a fair amount of experience as a researcher and doing other service-related work. My other faculty members at the University of Washington that are involved in the PEHSU include Dr. Sheila Satyanarayana. She is a reproductive environmental health specialist. She is particularly interested in exposures during pregnancy and early life to endocrine disrupting chemicals like phthalates, bisphenol A. We also have Dr. Esther Min, who's a community partnership specialist at the University of Washington. And I share the leadership of the PEHSU with Dr. Mary Kate Cardone, who's trained as a nurse practitioner and really oversees our consultation service. We also have nurse consultants, Margaret Willis, Christy Lanciati, Christy is actually down in Oregon. So she's housed at Oregon Health Sciences University. And then we have an exposure assessment specialist, Maria Chong, and we also have, you know, administrative support. But in that little box there, I want to highlight, in the last couple of years, we've started a scholar program where we're involving and engaging other professors early in their career at University of Washington to become experts in pediatric and reproductive environmental health. And so you can see the range of specialists that we're working with now, from the School of Nursing, pediatric pulmonology specialty, pediatric gastroenterology, OB-GYN, and urology. So we really comprise a lot of expertise in kind of integrating what we know from a medical community side into how it translates to public health and environmental health in communities. So who do we serve? We serve everybody. We really try to focus a lot of our effort on clinicians and healthcare professionals because they don't learn about this in medical school or other types of training into various careers as nurses, medical assistants, nurse practitioners, even public health professionals. The concepts of children and how children are especially vulnerable just is not integrated well into our curricula. So we feel like it's really important to serve that audience. And we're sort of specially equipped to deal with that nexus between medicine and public health on these issues. But we also get queries and requests from community members, community organizations, families, and the media. And in addition to just whatever question comes in and trying to provide expertise or information on where more information or resources might be available, we also get involved in special projects, and sometimes these come out of these connections with our clients. So I just want to give you a few examples of some of the sort of special projects that we are get involved in. And these are things that are pretty active right now. And we've been around for many years, as you see. So we've been involved in all kinds of things. But we have a lot of emphasis on lead exposure in children. It unfortunately is the gift that keeps on giving. So even though we've had a lot of regulatory action to reduce lead in the environment, we still see overexposure to lead in children. And we find that the clinical community is not well versed in what their role is in helping to address this issue. So we do a lot of showing up at clinical practices, doing presentations to, big settings for doctors and nurses and other medical personnel, so they know, what they're supposed to be doing, how to do it, and we support them in doing it. And we also support community partners in their role in helping communities be aware of these issues, going to community events, helping do actually lead testing in community events, which is something I'll be doing tomorrow. Another big emphasis area for us in the last couple of years has been the topic of relationship between environment and pediatric cancer. And we've been focusing on educating community health workers who are out in community as health promotion specialists on all kinds of topics, but often don't have a lot of background in environmental health. And so this has been a specialty interest of the ATSDR and CDC. So we've been helping get that word out through the community health workers. For this group, in terms of water and fish, we are actually just at the beginning of working with some partners down in Oregon at their health department on thinking about guidance for healthcare providers around contamination of fish with PCBs and what that might mean for breast milk and breastfeeding safety. So we're just at the beginning of this, but that's an excellent example of how we interface, with public health. They're doing their work with the risk assessment and components of reaching communities, but they would ask for us help to reach the clinical audience. I mentioned I have a lot of interest in wildfire smoke. I do research on that, and it's of course a really important issue in the Northwest. And recently, we've been developing a lot of materials and resources for clinicians to educate their patients, to work with their patients to protect them and prevent exposure to wildfire smoke. And the perfluorinated alkyl substances have been a lot in the news in the last several years. There's been a lot of interest in trying to, again, connect information to communities that need to know. And so we've developed materials in partnership with health departments and our national network. What do clinicians need to know? How can they talk to patients about their concerns with PFAS or when they find out that there's PFAS in their drinking water, as well as showing up to community meetings organized by community organizations to answer questions for communities that are affected, et cetera. So it gives you a little flavor of the kinds of things that we get involved in. And this is just, again, a picture of a lot of the organizations that we have worked with, continue to work with throughout the Federal Region 10. And it's how we can really do what we do, because we are pretty modestly funded. And a lot of what we do is because we can leverage these partnerships and work together on shared concerns. So I will stop there and leave it open for any comments or questions. But most importantly, hopefully you have a little flavor of who the PEHSU is and what we do, so that you can think about opportunities to connect with us in ways that might facilitate the work that you're trying to do. So thank you.
Thank you, Dr. Karr. That was wonderful. And also, I love the last slide showing how the constellation works. Does anybody have any questions for Dr. Karr? Bob?
Yeah, Dr. Karr, thanks very much. That was great. So the way you presented it was these very sort of topic-specific exposure areas. How do you in the medical profession deal with additive effects? So each one of these you could see being subtle air quality, subtle lead, so on and so forth. How does the medical field embrace that and educate people on that?
That's an awesome question. I'm really glad you asked it. And I think what I like about being involved in this as a clinician is I think we are in a great position to think about things as the big picture. So when we're talking, for example, when I'm teaching clinicians about talking to patients who are concerned about chemical X, and maybe it's, we talk about what we know about that chemical and the health effects, but we always talk about, for example, say the health effect is something like neurodevelopmental health. That comes up a lot with many contaminants. And as we're talking about that relationship, we put it in the context, well, what else do we know affects kids' neurodevelopmental health? And, what are the risks as well as resiliency factors? So, yes, oh gosh, it's not great that your kid was exposed to lead. And there's nothing we can do to change that historically. But we know that, when we think about lead and neurodevelopment, we know there are a lot of other pro-neurodevelopmental things that you have control of as a parent, as a community, we as a society. So what are those things and what can we do to, for that individual child or communities of children to support their best neurodevelopmental health outcomes?
Thank you. Heather, and then we'll move on.
Okay, real good. That was awesome. And I hope you're tracking the lead in cookware. Elmer, who's on this Zoom, was with us in Kent testing communities' frying pans that they were bringing in. And we did find lead in some of the frying pans that people are using. These were recent refugees. But my question is, when or if or can you expand your scope of work to include the issue of the microplastics impacting human health? Is that something that's on your radar, of something you might be able to include, or is it, funding dependent, or, that kind of thing?
Awesome question again. I love it. It's totally on my radar screen because I'm an environmental epidemiologist, environmental health specialist, and it's on the screen of those of us who think about contaminants in the environment. It's the emerging sort of hot topic. And Dr. Sheila Satyanarayana, who I mentioned is affiliated with our PEHSU, among us is probably the most expert on that. As you probably are aware, it's emerging. The science is still developing. The human epidemiology is pretty limited, but I do think there's a real need for more sort of translational materials and information to help people think about that and all of us to think about, sort of what we can do at an individual level, again, policy level, program level. And I will say our PEHSU does not currently have a microplastics, fact sheet or frequently asked questions. And it's something that I think is on our radar screen, and we're thinking about, developing. But I think it's a really important topic, and we do a lot of that. We're comfortable dealing with the uncertainty. I think that's part of why things aren't there. People have a hard time developing these materials sometimes when we don't know enough, but I think we do know, enough now to be able to at least say something that might be helpful and useful. And so I would say stay tuned, but yeah, it's kind of a matter of picking priorities and getting the person power to make it happen. But hearing you plug for it, is always good for us as well. When we get, queries and or hear from organizations saying, gosh, we really feel like we need, some information on this. That's what motivates us. So.
All right, this conversation will be continued for sure. Dr. Karr, thank you so much. We have to move on, sadly, but if you have additional questions, please put them in the chat and we'll make sure that we follow up. Thank you again.
Yep.
Happy cycling. All right, next we are going to welcome Maureen Swanson from the ARC and Project TENDR. Welcome, Maureen, and thank you for joining us from the East Coast. You can share.
Happy to be here. Should I just go ahead and share my screen now, Heidi?
Yes, that would be great. Thank you.
Okay. I will
Let us know if you have any problems.
I think we're good. So I will be talking about Project TENDR's work on kind of bringing together the science on what we know so far about microplastics and the toxic chemicals in plastics and impacts on children's developing brains, and a little bit of how we work on that, and kind of where we see there to be opportunities. And thank you for this opportunity today to talk with you all. So Project TENDR stands for Targeting Environmental Neurodevelopment Risks, and we're an alliance of more than 50 scientists, health professionals, and environmental health advocates who came together in 2015. Dr. Irva Hertz-Peixoto, who's an epidemiologist at UC Davis, and I co-founded Project TENDR. And I am not a scientist. I have a background in environmental policy and advocacy. And our scientists in Project TENDR are at universities all over the country. And they are epidemiologists and toxicologists, exposure scientists. We have health professionals who are, I think Dr. Mark Miller, I think he's still involved with the PEHSU in out of UCSF. So we have health professionals who are at health systems and university hospitals. And also we have representatives of National Hispanic Medical Association, ACOG, the Association or the American College of OBGYNs, the National Medical Association, and then our health advocates, our environmental health advocates are they are policy and legal experts at NRDC and Earth Justice and EDF, and also at the Alaska Community Action on Toxics, which some of you are familiar with, I know, and at Healthy Babies, Bright Futures. So we have those three constituencies, but there's a lot of overlap. And our purpose is to protect children's developing brains from neurotoxic chemicals and pollutants. And we do that by bringing together, synthesizing the scientific evidence and publishing journal articles on the scientific evidence along with policy recommendations. And then we seek national media coverage, national, state, local media coverage on our issues, on our publications and neurotoxic threats. And we take our science and recommendations to decision makers. We do a lot of meetings with agencies. We help our scientists plug them into opportunities to provide testimony and public comment letters. We also do amicus briefs recently to help inform court cases. So all aimed at bringing the science to influence and inform policy change. And just quickly, we operate on a consensus basis as far as making decisions about what we work on together. And so a little bit of background, at our fall annual meeting in 2022, we made a consensus decision to engage on plastics and neurotoxic chemicals, that should be a priority area for Project TENDR. And we got started. We wrote and submitted a comment letter on EPA's proposed strategy to address plastics pollution. And as some of you are aware, those comment letters go into the public docket, and they can be helpful to the agencies, but they're also helpful in getting that scientific evidence into the public record so that it can be used in court cases and in other ways by NGOs. We then, the next fall, 2023, we deliberated and further decided to write a plastics briefing paper. And this was all informed by Pam Miller's leadership. She's a wonderful, Pam's amazing. And she's a leader within Project TENDR. And so she was urging us to put something together quickly to help inform the global plastics treaty negotiations. And I should say our plastics work builds on our long time work around phthalates, flame retardants, and other classes of neurotoxic chemicals that are part of plastics. So I will, and there's that on the right, that is our, the cover of our briefing paper. That's a little bit of a different effort for Project TENDR. We typically write papers to be published as a first step in the scientific and medical journals. But in this case, we wanted to pull something together quickly. So the briefing paper or briefing report is fully referenced, but it is not in the peer-reviewed literature. So this I wanted to show just quickly to get a sense of, this is a subset of Project TENDR, and these are the authors of the plastics briefing paper. And in particular, Carmen Marcet, who's an epidemiologist at Emory University, and Beata Ritz, who's an epidemiologist at UCLA, were lead authors for us on our briefing report. So we issued the plastics briefing paper in April 2024, just before the 4th treaty negotiating session. And thanks to our partners in ACAT and the IPEN, International Pollutants Elimination Network, Endocrine Society, we were able to disseminate our paper to every single delegate to the global treaty negotiations, as well as to within the US State Department, EPA, and partner NGOs internationally. We held a UN-hosted webinar for more than 200 people in April 2024, including some of the treaty delegates. And we worked again with those organizations you see there to further share and promote the briefing paper throughout last year. And it was disseminated again right before the 5th treaty negotiating session in November. So that culminated in the TENDR briefing paper, contributing directly to new treaty provisions that for the first time address health and chemicals. And we were, the International Scientist Coalition involved with the treaty let us know that. So what does the, and I should say there's another treaty negotiating session, as many of you might know, in August in Geneva. And we have submitted a commentary to a scientific journal that we're hoping might be published in time to get that to the delegates as well. It might not be, but we'll see what comes out of that supposedly final negotiating session. So we are exposed to microplastics and thousands of chemical additives. And the reason I left the line through plastics fragments is we in TENDR, like a lot of people, are focused on trying to figure out how to best communicate this science around plastics. And we used plastics fragments and plastics particles in the briefing paper. And in the year or so since then, we've been advised by a number of communications specialists that microplastics is resonating with the public a lot more. So now in our commentary and in our other work, we use microplastics. But I know a lot of you are engaged in exactly that same effort, trying to figure out how best to communicate the science to the public. So there are classes of known neurotoxicants in plastics, including phthalates, bisphenols, and flame retardants. And then there are other categories of likely neurotoxicants where the science is more emerging, in part because some of those chemicals are replacement chemicals, so they haven't been well studied. including the other organohalogen flame retardants, organophosphate flame retardants, chlorinated paraffins, and PFAS. And plastics, as Dr. Karr noted, the human epidemiology is still limited on microplastics and health, but we know a lot. There's decades of evidence on some of those classes of chemicals that are used in plastics and leach out of plastics. And there's just a quick slide. This is a slide from Dr. Marcet, Carmen Marcet at Emory University, who I mentioned is a leader of our plastics work. You can tell Carmen's slides because they're so much more interesting to look at than mine. But it just shows some of the fragmentation. And this on the left side, that's a coronavirus. So he was explaining that some of the nanoplastics are as small as the coronavirus and can similarly enter cells very easily. So microplastics and the chemical additives are inhaled, ingested, and absorbed from diet. Diet is a major source. These chemicals aren't bound to the plastics, and the microplastics themselves also are breaking down and in food and drink. They're in dust. They leach from plastic products, including flooring, building materials, electronics, and they concentrate in dust. And then the incineration of plastics that include chlorine or bromine can result in neurotoxic chemicals. Children are more highly exposed than adults. They spend a lot of time on the floor. They put their hands in their mouths and objects, and they breathe, eat, and drink more relative to body weight than adults. And that's something that that was one of the first things I learned when I entered the environmental health field. But and it sounds basic, but I feel like that's gotten lost recently. I think we need to start sometimes by reminding people that children are more highly exposed and more impacted. So microplastics and their chemical additives are found in the placenta, in newborns' first stool, in breast milk, in infant formula. And we looked at one study out of Hawaii, and I know it's just one study, but it was so striking that the study found it was banked placenta samples. And in 2006, 60% of the placenta samples contained plastics. And in 2013, 90% of the banked placenta samples contained plastics. And in 2021, the researchers found 100% of the placenta samples contained plastics. And now studies in 2024 and 2025, of course, as we've all heard, have found microplastics in human brains. And those are small sample sizes, but I expect that there will be more studies finding plastics accumulating in human brains. This is Carmen's other slide. Again, he puts great slides together. But there on the right, you have some of the sources of plastics exposures and chemical exposures. And on the left, of course, you have an illustration of all the places that those plastics and toxic chemicals are found. Now, again, there isn't much research. It's new in part because we are just in the last 3 or 4 years able to measure microplastics in people. But we looked at some review studies that are finding that microplastics, as well as the chemical additives, can penetrate the cell and impair the function of the placenta. And that in turn can affect fetal growth, brain growth and development, behavior, motor function, learning, and memory. And babies with higher microplastics exposure have lower birth weight, length and head circumference, and lower APGAR score. And lower birth weight in turn is a risk factor for neurodevelopmental problems. Now, while the science on microplastics and health effects is emerging, we have decades of evidence for some classes of chemicals in plastics that show that prenatal and early childhood exposures are contributing to neurodevelopmental disorders and problems with child brain development, and neurodevelopmental disorders, including autism, ADHD, and learning disabilities. And that's true of the phthalates and especially the PBDE flame retardants, and to a lesser extent to the for the bisphenols and brain development. So for all of these chemical classes, they leach from plastics into food and dust. These chemicals are widely found in pregnant women and in infants and children, and they pass to the fetus via the placenta and to the infant through breast milk and formula. I'll just quickly look at each of these. The phthalates are everywhere. They're in food packaging and food processing materials. They're in personal care products and flooring and wall coverings. Our youngest children are the most highly exposed. Dr. Stephanie Engel led a team that just published last fall a study showing that 0 through 3-year-olds have the highest exposures to phthalates relative to all other age groups. And food and drink consumption accounts for more than 50% of exposures in the United States for all age groups. Prenatal exposures to phthalates are linked to impaired IQ and problems with behavior and attention. And there's some strong evidence linking phthalates specifically to ADHD diagnoses. Phthalates can also, again, Dr. Engel at UNC is using brain scans of children's brains, showing that phthalates can change the brain structure, including the brain matter volumes. And the brains are changing in areas with links to IQ, to intelligence and learning. And so the emerging evidence shows that the replacement phthalates are also harmful to brain development. You know, DEHP is a notorious phthalate, well-studied, highly neurotoxic, and the levels of DEHP are going down in people, but the replacement phthalates are increasing, and they may turn out to be just as bad. The flame retardants, the organohalogen flame retardants, brominated and chlorinated flame retardants, are the PBDEs, the polybrominated diphenyl ethers, are extensively researched, and they are associated with learning and behavior and intellectual impairments, and they can disrupt thyroid, which impacts brain development. And although the PBDEs are banned internationally and restricted from some uses in the United States, they've been found in recycled plastic products, which Washington State and University of Washington and Toxics Free Futures has led the way on that research. And then likewise, other brominated flame retardants and organophosphate flame retardants have replaced the PBDEs. But I was talking with Dr. Heather Stapleton at Duke, who's an expert on flame retardants, and she was telling me that, for example, Deca BDE, which is has been largely phased out, but is found in recycled plastics, so levels of Deca BDE are declining in people, but levels of Deca diphenyl ethane, a replacement flame retardant, are increasing in people. So again, it's a terrible problem. And the emerging evidence of course, everybody scrambles to try and study the replacements instead of our policymakers addressing these toxic chemicals as a class. And the replacements are also showing neurotoxicity. And then the bisphenols, the neurotoxicity of bisphenol A has been extensively studied in people and in rodents, and has been found to likewise, those prenatal exposures, as well as exposures in infancy and adolescence, are linked to deficits in memory and cognitive function. Human studies indicate BPA could contribute to problems with attention, autism, depression, anxiety, and cognitive disorders. And again, the studies that are starting to happen on the replacements are actually showing that some of the replacement bisphenols may have greater impacts on child brain development than bisphenol A. So we have a set of recommendations that we made in the briefing report, and then we modified, and we have included these recommendations in our upcoming commentary. And these recommendations are developed in consultation with policy experts at NRDC and at government agencies and at Earth Justice and EDF. And so we need to limit plastics production and start with single-use plastics, and especially packaging, which makes up 40% of plastics is packaging. And it's incredible to me that not only are we dealing with all of this plastics, that plastics pollution and plastics in products, but now companies are adding nanoplastics and microplastics to products. And so we need to ban those. And then our recommendations focused on reducing the toxicity of plastics is we advocate for banning classes of toxic chemicals used in plastics, starting with bans right away on plastics that come into contact. Bans on the neurotoxic chemical classes in plastics that contact food. And then phasing out production and use of the most toxic plastic polymers, which include PVC and polystyrene, which are both carcinogenic. We'd like to see government standards set to aimed at the elimination of plastics from drinking water and the prevention of toxic plastics, so-called recycling, and then ensuring that the disposal and recycling of plastics does not result in releases of toxic chemicals and microplastics. And there we have some recommendations around transparency and information, including testing drinking water for plastics and making the test results public, and adding microplastics to the United States National Biomonitoring Program, the NHANES. And I will note that NHANES is part of CDC and part of the National Center run out of the National Center for Environmental Health, which is being dismantled. Apparently, the NHANES program is supposed to be moved under the Health and Human Services structure, but we know that NHANES is threatened, and we're not sure where it will end up. And then I'll end with we have an ongoing plastics initiative. We have prioritized work on plastics moving forward. So we're doing presentations everywhere we can, and we are so appreciative of these opportunities to engage with folks and learn from others, as well as sharing our science and recommendations. We just did a presentation on plastics and children's health in front of the Society for Environmental Journalists, and that was fascinating to hear what journalists are trying to understand about about plastics exposures. And then we have new work that we are starting and that we're also applying for funding for some new work to on our messaging and communications, because we can certainly keep improving that, especially in the context of the MAHA movement. And we may be holding a congressional briefing and Hill visits this fall, aimed at helping people understand what the science shows about why these exposures matter to people's health, and to highlight the need for a policy response, policy action. You know, a lot of the journalists at the Society of Environmental Journalists Conference asked about steps individuals could take to protect themselves or their families. And that's important, but that's not going to get us out of this mess. We need policy action. And I'd be happy to answer questions and have a discussion. Thank
Maureen,
you.
Thank you. That was both amazing and highly disturbing. We are running a little bit over, so we have time for one question, and then please put your questions for Maureen in the chat and we will get them answered after today. Does anyone have a question they'd like to ask? Maureen, thank you also for clearly laying out the policy action plan. That's very helpful.
Thanks.
It was great. And thank you for joining us. I hope you stay on for a while.
Absolutely. Thank you.
All right. Thank you. So next we are going to hear from Dr. Susanne Brander from OSU, who has been thick in international affairs. And I enjoy seeing Dr. Brander on LinkedIn quite a bit. So welcome, Susanne.
Good to see you this morning. I'll go ahead and share my screen. Are you seeing the right set of slides here?
Yes, it looks great. Thank you.
Okay, wonderful. Thank you.
We are seeing your speaker notes.
Oh, okay. I'll swap displays. How's that?
Perfect.
All right. Thank you so much, Heidi, for inviting me and others, Dr. Elise Granick and Dr. Stacy Harper, to be represented in this talk. So I'll be talking, kind of giving a general update on microplastics, kind of where we are with research that has been done at Oregon State University as well as at Portland State University. I will say it definitely takes a village in terms of compiling resources from different granting agencies, different types of expertise from different laboratories, and of course, all of the wonderful students and postdocs and undergraduate researchers and technicians that we've had working with us over, or at least for the Pacific Northwest Consortium, which I co-lead with Dr. Stacy Harper for the past about 5 years now. So to start off, and others who've given talks this morning already have alluded to this, that the microplastics problem is something that, it's everywhere we look. The more we study it, the more we realize there are things we haven't thought about yet. There are, there's information hiding in the corners. We're finding plastics in very pristine areas. So it's really an everything everywhere all at once issue. And we know that both organisms and humans are exposed via a variety of different pathways, and they are where we haven't looked yet too, I'm sure we'll be finding in the coming years. And just like any other environmental contaminant that has been studied, from lead to PFAS to PAHs and bisphenols, we're all exposed, whether we are human or whether we're wildlife, in different ways. So Today's talk is going to talk a lot about marine organism exposure and freshwater exposure because that's my main area of expertise. But we've also begun thinking about how to translate this information to what's happening for human health, for mammalian models, and we're starting to see some similarities in responses between different taxonomic groups. But if you're thinking about this from an aquatic organism perspective, a lot of the work that's been done over the past, at this point, 10 to 15 years, there's been heavily focused work on microplastics, has been happening across biological scales. And we're starting to see that there are effects right from the molecular level all the way up to potentially the ecosystem level and beyond. There are groups even thinking about how microplastics and plastic pollution contribute to the near exceedance of planetary boundaries. So really thinking about this holistically across different biological scales, and also thinking about how plastic pollution is interacting with other threats to the planet, such as biodiversity loss and climate change, is incredibly important at this time. But coming back to the ocean and thinking about where most of the microplastics we are seeing are coming from, textiles and tires, at least at this point, are estimated to have the largest presence in terms of particle number for microfibers and in terms of mass for car tires in the ocean. This is likely similar for freshwater. Terrestrial ecosystems have not been as extensively studied, but we know that tire particles and microfibers and other plastic types are of a big concern to terrestrial food webs. And there's especially been a lot of new attention paid to contamination of our food supply and agricultural plastic contamination. But in terms of what we are seeing in freshwater and marine ecosystems, almost any fish, any sediment sample, water sample we pull out and analyze, we're seeing mostly microfibers. If it's sediment near an urban area, we're seeing a lot of rubber particles that are likely tire wear from cars. So today I'm going to split this talk up into 3 different parts. I'm going to talk first about exposure and kind of where we're going with exposure, what we're finding with a few selected studies in the aquatic environment. I'll talk about responses in fish and then relate that to findings from other groups in mammalian species and thinking a little bit about it, even extrapolating that to human health. And then we'll finish up talking about some mitigation studies that are ongoing, as well as regulation, and we'll span that out to the global level and talk a little bit about what's happening with the treaty to add on to the previous talk, which got into the global treaty on plastics a little bit as well. So in terms of exposure, this has been quite a journey for our lab and for others in terms of figuring out what the best way is to figure out how many microplastics are in a diversity of different samples. And so early in the field, people were mostly looking at water samples or maybe looking at macroplastics in the guts of larger animals. Now we're at the point where we're even trying to get to the sub-micron level in terms of trying to figure out what is present in terms of microplastic particles as well as other materials. And so what our lab and many other labs do is we go through a series of steps which take quite a bit of time and concentration and effort, but it involves isolation and dissection of lots of different materials. At this point, we're working on everything from zooplankton to bird scat to biosolids from wastewater treatment, digesting those samples in a variety of different ways to get rid of the organic material, doing inspection and measurement, what is our size range, what types of materials are we finding, and identifying the material type using spectroscopy approaches, such as Fourier transform infrared spectroscopy. These methods have advanced quite a bit over the past decade, and at this point, the state of California is even trying to put forth some standard protocols for sampling for microplastics and also for the digestion of complex samples like sediments, which we've been involved in. So just to give you an idea of what my students spend a lot of time looking at during the day, and students in other labs working on microplastics. It's a lot of very detailed and time-consuming work, and measuring these particles using programs like ImageJ or sometimes approaches like Nile Red to make things go a little bit faster. But really, at the end of the day, to get accurate results, sometimes you're needing to characterize particles individually in these samples that can take a long time to process to begin with. So a little bit of a snapshot of some different studies that our lab has been involved with or has led. Recently, we were able to collaborate with a lab here at the Hatfield Marine Science Center, where I am based, at NOAA, which has been collecting lanternfish, also known as myctophids, for a number of years. And so they had a freezer full of fish that we were thankfully able to access. And former master student Olivia was able to look at the contents of their guts and found that, not surprisingly, most of what they had ingested were fibers. About a third of the species contained particles. And if you scale that up, it doesn't sound like a lot, a particle or two per fish and a third of the fish. But if you scale that up to what, say, a salmon would eat or a tuna would eat, if they were preying on myctophids, it could equate to millions of particles per year. And so this was an important study also because we were able to see that the myctophids closer to the coast contained more microfibers and microparticles. And it should be noted that a lot of these microfibers were not necessarily synthetic. Even after we accounted for background contamination in our laboratory, which is standard now, we did see a lot of cellulosic fibers, which indicates that those cellulosic fibers, as we know, shed more easily. They're coming out into effluent and via biosolids from our laundry, and a lot of them are likely chemically treated, which makes them more resistant to breakdown. So just something to think about, that when we're thinking about microfibers, we're not just thinking about polyester and nylon, we're thinking about everything we wear. So we also did a study in collaboration with museums at Scripps and at the University of Washington. And Olivia was able to get samples dating all the way back to the 1960s, all the way up to 2015. We were a little surprised that she didn't see as big of an increase in the number of fibers per myctophid with this decadal sampling, but she did see a nearly significant trend looking across those years. And of course, the other thing to mention here is that some of those fibers may have degraded, and this is going to differ depending on the species you're looking at too. So more research needed, as always. Kind of scaling this up to coastal food webs, we've also looked at the accumulation of microplastics by prey that are quite important to iconic species like the Pacific gray whale. So this was in collaboration with the Marine Mammal Institute, which is also based here at Hatfield, and using data that they had on the dietary needs and consumption by pregnant and lactating gray whales. We looked at zooplankton and what they were internalizing. And we estimate that based on what the zooplankton species these whales are feeding upon, and that they need millions of them per day, that your typical Pacific gray whale could be exposed to over 20 million particles per day based on their ingestion of these different zooplankton species off the coast of Oregon. And so all this work was done right off the coast of Newport, kind of in that region. So another study that came out late last year, and this is a study led by Dr. Elise Granick and her student Summer Trailer from Portland State University that we collaborated on, is that we know that these smaller species are taking up these microplastics. But does it really matter to these larger commercial fishery species? A lot of, I think the thinking before this study came out and other studies like it came out was that, well, they're ingesting these particles, but then they're excreting them, right? And we are not eating the gut. And so why should we be super worried about what these larger organisms are ingesting? But it turns out that if you look just in the edible tissue, the muscle tissue, that you're finding some of these larger particles, you're finding microfibers in the muscle tissue. This is after, of course, like I said before, controlling for background contamination. These were fibers that were identified using the FTIR process, digestion process that I referred to before. And so this really opens up a lot of questions about translocation. How are these fibers getting from point A to point B? They're either moving from the gut into the muscle tissue by sort of crossing those epithelial barriers, or they could be inhaled, respired, right, while the fish is moving water across its gills, and they could be somehow moving through the body that way. More likely, it's coming from the gut. And a review we published also last year proposes a few different mechanisms based on a number of different papers for this translocation. Things like endocytosis, M cell uptake in the gut, persorption, or even kind of moving through these tight junctions, especially if the gut has been damaged and is leaky, are all possibilities. And there's a lot of debate as to how small the particle needs to be. In fish, a paper from a couple of years ago estimated that those particles could be as large as 80 microns. In humans, a lot of the literature supports a smaller size that is below 10 or 5 microns. But either way, these microfibers are quite narrow in one dimension. So in the more narrow dimension, they can be between 8 and 14 microns wide. And so if they're crossing in that dimension, it's possible that they're able to be translocated via one of these mechanisms shown here. But of course, more research is needed on this as well. And it's a bit concerning, not just for fish, but also for other vertebrates. We need to understand how this is happening. So I'm going to talk a little bit about responses now. And again, most of our work over the years has been on aquatic organisms. So started out when I was based in North Carolina, thinking about the tiniest of organisms like these tintinnid ciliates, which are only about 100 to 120 microns in size, but they're at the base of freshwater and marine food webs. They are consumed by larval fish. We found that they readily take up small plastic spheres that are about 10 to 20 microns in size, and then can transfer those to larval fish at higher rates than the fish will ingest particles on their own. And so this led to work in collaboration with Dr. Stacy Harper and with the Pacific Northwest Consortium on Plastics, where we really wanted to get at the particle questions, because back, when we started this work in 2019 or so, end of 2019, most studies were being done with polystyrene spheres, including studies in fish. This is still an issue for studies in mammalian models. You can easily purchase a little vial of polystyrene spheres from Thermo Fisher. They're labeled, you know what size they are. It's easy, right? But the problem is that we were seeing that we never or rarely found a sphere in an environmental sample. So if we really want to understand how humans are being affected, how wildlife are being affected by microplastics, we need to use particles that look like what is actually found in the environment. And so in collaboration with Stacy's lab, she's in engineering and toxicology, I'm in fisheries and wildlife, she was able to come up with a method to cryomill particles that were more realistic, that we could use in a variety of different exposures, with her lab focusing on freshwater organisms like zebrafish and Daphnia, which are pictured here. My lab focusing on coastal organisms like mysid shrimp, which are important prey for gray whales and salmonids, and silversides, which are an estuarine fish species used by the EPA for toxicity testing. So between our two groups, we were able to come up with methods for cryomilling a variety of different particles, from tire wear to polypropylene to even bio-based plastics like PLA. And then my lab has been developing a method using a cryostat to make polyester fibers and fibers of other material types that we now use in exposures. So not exactly the same as what you'd find in the environment, but closer than spherical particles that don't have the same complexity as the fragments and fibers and foams that we typically find in our sediment and fish and water samples off the coast, or really anywhere at this point. So today I'm going to focus mainly on tire wear and polyester fibers because those are some of the most frequently detected particles. So some studies that we've done, early studies that we did with fibers, comparing them to fragments of the same plastic type, indicated that fibers probably were causing more stress in comparison to other particle shapes. And so this is a study we published in 2021. We looked at effects on fish respiration of exposure to fibers versus exposure to fragments. And again, this is the same plastic type. And we found that only the fibers caused respiratory stress. And so because of this study and other studies that had been done at the time, we decided that this was going to be one of the main particle types we would focus on, including its frequency of detection. And then, of course, tire wear has been another big area that we've been working in. Clarissa Raguso, who was a PhD student in my group, is now a postdoc at PSU, was looking at tire wear mixtures. And so another kind of jump in the field over the past couple of years is that those of us working with tires have been able to strike up a collaboration with the U.S. Tire Manufacturers Association. They are very interested, of course, in trying to figure out what's going on with chemicals like 6-PPD, which I'm not going to get into today, but they're also interested in particle effects. And so we were able to use these realistic mixtures that they've made up from tires that are used in different parts of the country, so trucks, cars, buses, and create a mixture for exposures to some of the organisms that we work with. And what Clarissa found was really interesting, I should say, Dr. Raguso now, found that the weathered particles were, in this case, potentially more damaging because the organisms, both the fish and the shrimp, were taking them up more frequently, and that was directly connected to reductions in growth rate. And so they eat more of the weathered particles, and then their growth rate was more affected by the weathered particles. And I think this study is important, the results are important, because what it indicates is that it's not just about the chemicals, it's about the physical toxicity and the fact that these particles are taking up space in the gut and then causing responses in those organisms that could affect their likelihood of survival. We also look a lot at behavior. I'm not going to spend too much time on this, but I think it's interesting that we're looking at the potential neurotoxicity of particles and plastic chemicals in humans, and we're also definitely seeing neurotoxicity in aquatic organisms. So a pretty standard assay in smaller larval fish and invertebrates is this Noldus ethovision assay, where you can measure things like swimming behavior, time spent bursting, cruising, freezing. You can even kind of measure how often they're hugging the wall, which indicates potential for anxiety. This is standard, it's been used for lots of different chemicals. And what we see in fish, larval fish, and in juvenile invertebrates is that the tire particles have different effects based on which species we're working with, and also based on whether they are weathered or whether they are pristine. So we really need to have a better understanding of how this is affecting the behavior of aquatic organisms and how that translates potentially to neurotoxicity in other species. There may be a common mechanism here. We see effects on reproduction. So briefly in Daphnia, which are a great model for studying reproduction because they can be reared across generations. They have a very short lifespan, which makes those studies rapid. We found that reproduction was affected by exposure to sublethal concentrations of tire. So the number of neonates was reduced. And this was also shown in the offspring, especially of the nanotire particle exposed parents. And so this is potentially a cross-generational issue. So those are our results on tires, just kind of in a nutshell. Going to talk a little bit about microfibers now. And kind of getting back to that idea and think about the graph that we showed with the myctophids and that a lot of what they were ingesting was cotton. If we bring that back to the lab and look at what happens in a controlled study where you're exposing organisms to cotton, polyester. We also did polypropylene, although the data aren't shown. You see that the cotton can also have an effect on growth, just like the synthetic fibers do. So again, it's really getting back to this idea that it's about the microplastics, but it's also just about the volume of materials we are putting out into the environment, and that some of these materials may be natural, but they've also been treated with pesticides and lots of other things that make them more persistent and potentially harmful to some organisms. So for both the cotton and polyester, we saw reduced growth in the mysid shrimp. This is a lot going on in this slide, and I'll briefly explain. We've moved from that study now to a current study where we're trying to look at both the physical and the chemical toxicity here. And so using the same species, Lauren, who is a PhD student in our lab, is comparing untreated fabric with ozonated and chemically softened to try to get at what the combination of chemical and physical effects are on these species. And she's finding some interesting results, and maybe not surprisingly, she's finding that the chemically softened fibers that have a range of different chemical mixtures applied to them have more effects on organisms' behavior than the naive fibers do. And so a lot more work to be done here, and we're also going to have some chemical analysis done on each of these treatments as part of her work. And kind of zooming out to the bigger picture, of course, we have all these individual studies on individual particle types, and while that's interesting, maybe the more important question is, how do they compare with one another? And so when we look at our data from different plastic types like fibers and tire particles, and here we even have PLA, which is a bio-based plastic, and you compare across all of those in larval fish, you see that the microfibers have the biggest effect on growth here. And interestingly, the microfibers are also causing more changes in gene expression than some of the other particle types. And what we found from this study as well is that the microfibers, when they are weathered, as you can see in this image here, they start to create nanoparticles. So this is a polyester microfiber. And then if you look at this graph here, and you see you've got the nano PLA here, nano tire particles, weathered microfibers, those three are often causing the same types of changes in gene expression in genes related to things like muscle functioning, cell signaling, and cardiac muscle function specifically popped up a couple of times. So does that relate to what's seen in humans in terms of some of those epidemiological studies linking cardiac events to microplastics? We don't know, but I highlight that. I highlight things, I realize I'm running out of time, things like studies that have shown that similarly in mammals that are exposed to microplastics, you see restrictions in growth, you see that there is disturbance of the muscle system, disturbance of metabolism. I think we're really getting to a point in this field where we can start kind of connecting the dots between these different types of organisms rather than drawing, a silo around the aquatic and a silo around the human or mammalian responses. I think they are united like they are for other contaminant types. And there was a review that came out just a couple of years ago and in 2025 that kind of highlighted this, showing that you see these effects in fish such as oxidative damage, tissue damage, neurotoxicity. And then over here for humans, you're seeing even in early studies where a lot more research needs to be done, some of those same responses are showing up in humans. And again, I think, you can go back to Rachel Carson where she saw effects on DDT in birds, and then years later we found that it was also a carcinogen in humans. I think in some ways we're headed in a similar direction here. We just don't have all the data yet. So I'm going to finish up with mitigation and regulation, and I'll keep this brief. Some things that we're now doing in collaboration with Portland State University. They are leading some great studies funded by National and Oregon Sea Grant to think about how to mitigate things like fibers, which are being produced in large volumes, by laundering, for example, and other activities, or thinking about how to mitigate tire particles. So the main particle types we're finding in our samples. And we know there are terrestrial sources. We know those are getting into aquatic environments. We know that wastewater treatment plants don't have good ways of mitigating these particles without really completely overhauling their design. We know that things like biosolids are heavily contaminated with microplastics and microfibers. So what do we do about all that, right? It can't just be all bad news. We have to think about solutions. And so there are these two studies where mitigation approaches are being explored in Oregon and Washington, both by putting filters on washing machines. And the newer study will also look at dishwashers and dryers. And then looking at stormwater intervention and putting stormwater mitigation devices, capture devices, to see if we can capture tire particles and other non-point source plastics that are getting into our stormwater system. So I'm going to finish up thinking about global policy. And as was mentioned by Maureen, I believe, we have an upcoming INC 5.2, which is supposed to be the final set of negotiations happening in Geneva this August. I plan to be there as part of the Scientist Coalition, which I've served on for a couple of years. I lead in areas like chemicals of concern in plastics and thinking about microplastics and responses in organisms. So we'll be there to share science-based information with delegates. We produce policy briefs. We meet with member states prior to negotiations to try to convey the latest science. And really, there's a lot to be hopeful for. There is a set of over 100 countries calling themselves a coalition of the willing that are really wanting to move towards more ambitious global policies and globally binding regulations. And so we'll have to see where this goes, because there are also, of course, powerful countries that are not in alignment with these same ambitions. And so that will play out in August in terms of what the hopefully final conclusion of these negotiations is. And so I'll finish up there for today. And this is just an image from the last INC where the delegate from Mexico read out that list of, I think it was 107 different countries that are in support of an ambitious treaty. But to summarize sort of where we are and future needs, we know from the work that we've done and from work from other labs that particles are becoming more toxic as they decrease in size. Fibers seem to be more problematic. We need more research on that. And growth, reproduction, behavior, and gene expression are impacted across taxa, not just in fish. We're seeing that in humans. We're seeing that in mammalian species. Species sensitivity can differ, but there are these kind of broader trends. Translocation is a big concern. We need more work. We need standard bioassays. We need multi-generational studies and better analytical approaches to really track those much smaller particles, as well as mitigation approaches. A lot of what we're talking about right now are downstream mitigation approaches, but what the treaty would provide in its most ambitious form would be upstream approaches to source reduction of chemicals, regulating chemicals by group. And that's really where we need to go in the long term if we're going to tackle this enormous challenge. So I'll finish up there, and again, we'll acknowledge all of the wonderful people I've gotten a chance to work with, and thank you so much for your time and for the opportunity to present.
Susanne, that was amazing, and also one of the best visual presentations I've ever seen. But you obviously have a lot of practice and are probably very influential in your circles. I think we do have one question in the chat for you, and then time for maybe one more question before we go to break. And I'm sorry, but please, again, put your questions for Susanne in the chat and we'll circle back. Kathleen asked whether the picture with the Daphnia with the small plastic particles inside the animal, were those the spheres?
The picture of the Daphnia for the rubber study?
I think so. Kathleen, you can come off mute if you want.
Yeah, thank you. In your slides, you had a couple different pictures of the Daphnia and it had little black circles in the gut. Were those eggs or were those plastic nanoparticles or what were those?
So you were, I could go back to the slide if I had more time, but I think you were seeing two things. The things that were almost perfect circles were eggs, were incubating neonates, and then the black particles that were irregularly shaped were tire particles that we had exposed them to. So they were exposed to both micro and nano-sized tire particles, and the nano-sized had a greater effect on at least multi-generational reproduction.
Okay, thanks.
Thank you. And then Bob, one question, and then sadly we have to move on for now, but we'll have subsequent chats. There's a lot to chew on here.
Sure.
So to speak.
Thanks for great presentation. On the weather, you mentioned that it's a future goal. Have you started looking at the chemical constituents bound to the weathered particles, and what have the most of those chemicals been as you've looked at that?
Yeah, so most of our focus has been on the toxicity, on the biological responses. I'll be honest, most grants will cover one thing or the other, right? Unless you collaborate with a chemical and an analytical lab, it can be difficult to get detailed information. There is a really great paper that came out last year led by Miranda Jackson, who is at Oregon State and collaborates with Stacy Harper's lab. Their lab was at least able to do some chemical analysis on leachate from the tire particles, the CMTT particles that we used in our study. I didn't have time to present those data today, but it's Jackson et al. 2024. If you want to check that out, they found things like zinc is a huge problem for tires, as well as the other, typical suspects like the PPDs and the benzotriazoles. Also, a review we published led by Paul Mayer of the EPA goes into a bit more detail about the chemicals of concern with tires. And then we're working on the fibers. We should have data on that later this year.
Thank you. Thank you again. We're so grateful. We're going to move now into a 10-minute break, and then we'll have to discuss whether we have to truncate the community conversation just a little bit. But there's so much to follow up on that we're looking forward to having subsequent discussions because there's so much here. Dustin?
Yeah, thanks, Heidi. Yeah, we'll go into the community roundtable after this. I think there's enough room in the agenda. So I'll put up a reminder here for everybody to return by 10:50. And I think that's the right, hopefully that's the right screen. And then we'll join up with the community roundtable. So if there are things happening that you'd like to share with this group, that'll be your time to share links and whatnot. So thank you very much. Thank you very much to our speakers, and see you in 10 minutes.
And Dustin, please.
Through for you all the Stormwater Strategic Initiative investments that we've made in the last few years relating to the implementation of toxics in aquatic life strategies. First, I want to recognize our Toxics Pod and our Strategic Initiative Advisory team, who are really helpful in developing the priorities that we put into our investment plans. And those investment plans drove the RFPs that we put together over the last couple of years. So from this pie chart, you'll see that we've got almost $16 million invested in toxics reduction related projects, starting from 2022 up to some projects that we're still working on getting under contract right now. Each slice of the pie here shows you the different RFP categories and the amounts that were obligated for each of those categories. And also, I do want to make sure that we recognize that these are funds from US EPA and the Puget Sound Recovery Office through the Geographic Program Fund. So we really appreciate that, and especially in these days when all of that's a little uncertain. I'm just going to cover a little bit about each of the projects or projects in each of these categories. This addressing toxic hotspots impacts on juvenile Chinook was one of our recent RFPs, and is based on geographic priorities where our Toxics Biological Observation System, or TBIOS program, has been finding PCBs and PBDEs impacting health thresholds for the different vital sign indicators, the juvenile Chinook in this case. And so this is leading to some great work that is very geographically specific. One of those geographies is Commencement Bay, specifically the High-Labos, Cumming, Blair waterways. And the Port of Tacoma, along with the UW Tacoma, are working on a source tracing project together that'll look at and find sources of PCBs and PBDEs that are continuing to recontaminate a lot of the good restoration work that they've already done in those waterways, but that are continuing to not meet sediment quality standards. So that'll be a project to look for, and hopefully, kind of continuing and be following the example of the Duwamish waterway too, is a good model to follow. Seattle City Light is doing a project where they're able to sample for PCBs electrified transformers within the Duwamish waterway. They're using some new technology that allows them to keep the grid electrified while they're doing their sampling work, so that they can get that work done faster at less of an impact to their customers. Also, the Lummi Indian Nation is going to be doing some 6-PPD, copper, and other PCB work in Lummi waters up in Bellingham Bay, and that's a project they're working on with the USGS. So I'll just kind of move quickly here so that make sure we get time for our other two speakers as well. The Chemical Action Plan implementation started in 2023. Well, our RFP was started in 2023. We did another round of this that I'll talk about, but this is really about implementing actions that follow the recommendations in these chemical action plans for PCBs, PBDEs, PFAS, PFOA, and PAHs. So we're working with several different organizations around building materials and other types of houseware products that we can replace through these incentive programs to get rid of PFAS in homes. They do have an emphasis on working with underserved and vulnerable populations, which is great. And we've also got Zero Waste Washington doing toxic chemicals in durable products and helping provide local government grants to implement some of that work as well. The second round of Chemical Action Plan implementation was just awarded earlier this year. And we included the 6-PPD action plan in that round because it was finished at that time. So some of the projects that were, actually all of them in this category, this round, touch on 6-PPD in some way or another, looking at either currently installed stormwater conveyances to remove 6-PPD, or looking at community wetlands and how 6-PPD is affecting amphibians. And then also in the middle here, the projects is one that I'm really excited about, and touches on what we are really kind of talking about at the pod right now, which is Public Health Seattle King County is going to be looking at PPD, 6-PPD really, and PFAS in fish consumers. So that will continue to build on some of the work that they've been, that Seattle King County has been doing with fish consumers in that area, and begin to get data on human body burdens for these chemicals. The innovative BMPs category was awarded last fall, and these are really all stormwater BMP effectiveness monitoring studies. The city of Des Moines is going to be testing several different types of stormwater BMPs for 6 ppd removal as part of their redevelopment of the downtown and marina area that they're calling the Marina Steps project. We have Long Live the Kings continuing to work on the Ohop Creek stormwater treatment BMP that they've had there for a little while and going to be testing that for 6 ppd removal. Wazoo is going to be looking at existing stormwater facilities to see how they are, how effective they are at removing 6 ppdq. And Stewardship Partners is testing their adopted downspout BMP treatment system for 6 ppd removal, and that is pulling stormwater off of the Ship Canal Bridge in Seattle. So that's some pretty nasty stuff. These projects are all being developed and executed in a way that will help us include the data and their findings in future iterations of our stormwater manuals, so that we can be able to better enforce and require stormwater BMPs for 6 ppd purposes. This toxics reduction in local pre-treatment programs is one of the projects that we're working on getting under contract right now. And this is going to be a literature review and analysis of local pre-treatment programs across the country and potentially internationally, and compare those to what we've got going on here in Puget Sound, identify knowledge gaps, and put together a guidance document that'll be useful for Ecology's MPDS permit managers and for wastewater permittees and industrial stormwater permittees and industrial wastewater permittees that are secondary to wastewater. So we'll have some resources there to help them better reduce the load of toxic CECs that are going to wastewater treatment plants in the first place. We've got 2 rounds of CEC research. This first round was awarded a couple of years ago now and is looking at sampling and occurrence of 6 ppd and related chemicals, CEC mixtures and toxicity and modeling, which will be really interesting. And that is definitely one of the tougher questions to answer. So really excited about getting some data on that. And then I think this project may have already kind of started wrapping up, but the enhanced CEC monitoring data in the White and Puyallup River watersheds. I believe some of that is also going to inform the work in Commencement Bay. The second round of CEC research that was awarded this winter includes these projects here. There's some advanced wastewater treatment project. There's a look at the effects of antidepressants and SSRIs on Chinook salmon that Ruth Sofield at Western is leading. And then also some more characterization of 6 ppdq and other tire rubber derived chemicals in Puget Sound. Another one that the SIL is doing is, and this was awarded last winter, is piloting solutions for CECs and wastewater. We're recognizing that we need to understand the removal that current wastewater treatment technologies can provide for some of these CECs, and these are technologies that aren't required in our MPDS permits for wastewater. But as CECs become a more understood pollutant coming from wastewater, then these technologies are going to become very important. And so this was focusing on PBDE removals. And then we've got some projects that were awarded a few years ago now under our Public Roads Retrofit Planning RFP. And these include some projects that are going to produce web maps and other GIS decision tools and examples of studies for addressing toxics in stormwater. So that was a quick run through of all of those. I don't know if anybody has any questions. These are all projects kind of just getting started or in progress, so there isn't a lot of results to share right now, but I'm hoping that in the coming year or 2 or 3, as those projects are wrapping up, we'll have great things to share with you all on what they found.
Thank you, Dustin. Does anybody have questions for Dustin? And you could also just follow up with him directly. There's a lot going on. It's all great. It's been very gratifying. Great. We're very proud also of our finance team that does all of the really difficult heavy lifting behind the scenes. And yeah, we're just very pleased. We're sorry that we have to work on these things, but we're pleased at the progress that we're making. So
yeah. And I think we have like the Stormwater Strategic Initiative has like another $10 million or so in other projects related to stormwater, climate resiliency planning, stormwater parks, and a lot of other stuff too. So we're doing a lot of great work.
Technical assistance, model ordinances. Yeah, there's lots going on. Let's not jinx it. Let's don't talk about it. Okay, so should we move on? We're a little bit early, which is, you know, like a miracle and very unusual, but we could move on to Sandra Dorning if you are ready, Sandra. So Sandra is from the Puget Sound Institute and is the official keeper of all things unknown and uncertain, which she will tell you about. Welcome, Sandra.
Thanks, Heidi. I'm just going to share my screen really fast. Are you seeing slides or presenter view?
You're in presenter view right now.
All right.
Which I personally love, but
how about now?
That's good. Thank you.
Wonderful. Well, thanks, Heidi, Dustin, and everyone for having me today. My name is Sandra Dorning, and I'm a research scientist at the University of Washington Puget Sound Institute. I'm here today to share some work that we've been doing over the course of the last year and a half to support the stormwater sill by updating a toxics research agenda to inform the toxics and aquatic life implementation strategy. So I'm going to provide some brief high-level background just for those of you who aren't steeped in this world. Dustin already provided some really great background, so that's going to help me save some time here. And then I'll mostly focus on sharing the results of our process, which was identifying top research priorities for toxics in Puget Sound. So the context for the work that Puget Sound Institute does to support the stormwater sill is that we are tasked with providing technical support to the EPA's Puget Sound National Estuary Program. And that program really aims that recovery plans like the Toxics and Aquatic Life Implementation Strategy are informed by science in the recovery planning and actions that are taking place. So our role at Puget Sound Institute is in part to provide support by developing topical research agendas for the various recovery topics that we're working on in Puget Sound. And the idea is that by identifying research priorities in these research agendas, that these priority research needs or uncertainties, as we often call them, can then inform the research work that we do in the region to try to advance and improve ecosystem management. So the idea is to identify where knowledge gaps are really proving to be barriers to better recovery planning and decision making. Ultimately, the goal of that work is that eventually we'll identify priorities, we'll conduct research in collaboration with a whole host of folks in the region to advance scientific understanding of those questions, and that eventually those research findings can make their way back into planning and decision making. And that, of course, is not a passive process. That's a process that PSI also plays a role in terms of synthesizing scientific information and translating that for a policy and management audience. So I won't go into detail into the full explanation of this figure, but this is a schematic that we use to really describe the work that we're doing, kind of from the beginning at the top left, which is to identify where these scientific research needs really are for various topics, work to co-develop these research agendas with experts and practitioners, use those research priorities to scope and inform research work that isn't just conducted by us, but is broadly conducted by researchers throughout the region. And then we can bring new information into the decision-making space through a whole host of different communications products. So at the center of this process is a tool that we call the Grand Uncertainties Matrix, which I just wanted to highlight briefly. This is a spreadsheet database that Puget Sound Institute manages, and it's publicly available on our website. This database is filled with all of the research needs that we have collaboratively identified with experts and practitioners for recovery topics, including toxics in fish or toxics in aquatic life. So this screenshot just shows a couple of those from the original toxics in fish research agenda that was produced when the implementation strategy started back about 5 or 6 years ago. And so the work I'm going to be talking about today is our recent effort to update this database and update a research agenda for toxics now in 2025, given the new research and also new questions that have emerged in that time. So we undertook a process beginning last year to update a toxics research agenda, and it generally followed this schematic here. We started internally by cataloging research needs or research questions that we observed in regionally relevant reports, documents, including workshop notes from previous stormwater sill toxics workshops, and other sources, and put together kind of a raw catalog of all of those research needs that we observed. And then we refined that down a bit to produce a long list by editing for clarity, consolidating duplicates, etc. Through this process, we ended up deciding we needed to split our list of toxics research questions into 3. Microplastics and 6-ppdq and tire wear particles are 2 topics that were not captured in the 2019 iteration of the toxics research agenda. We could see that there were a lot of research questions about those topics, as may be obvious to all of you. And we really wanted to make sure that we drew upon specific topical expertise on those topics as we were refining and identifying what the priority research needs for those topics were. So we kind of pulled those out as separate lists, and then the remainder of our research needs were captured in what we call our vital sign list. So this list included questions related to legacy contaminants like PCBs, PBDEs, PAHs, as well as all other contaminants of emerging concern. And that was linked to our Puget Sound vital sign indicators and targets that Dustin briefly talked about earlier. So generally then we handled each list slightly differently based on the expertise that we drew on, but generally speaking, we took those long lists and worked with a small group of experts to refine that into a short list of the questions that really were clearly linked to the implementation strategy, were clearly barriers to progress within that recovery context. And then we brought that short list to a workshop last September where we asked folks to prioritize. So many of you were involved in that, and we're really grateful to all of you for your input and great feedback that we received in that workshop. We essentially asked folks to vote among the short list of which questions they felt were most significant barriers to progress for toxics in aquatic life. And so now I'm going to share the results of that process and the top priorities that we identified through that work. So for our vital sign list, this is a graph depicting the number of votes that each of our questions received. So pretty wide distribution, spanning all the way down to 0 for some of the really low priority questions, and the top priority question receiving about 16 votes. The top 7 priorities are listed here, and they include questions such as what biomarkers can be used to monitor effects of chemical exposure, what is the cumulative impact of toxics mixtures and other stressors? Are biosolids significant sources of CECs? What are the priority compounds in stormwater specifically? What are the primary loading pathways in Puget Sound? What are the cumulative effects of multiple types of contaminants on the Puget Sound species? And then where are the geographic priorities for stormwater retrofits? that are necessary to intercept road-derived toxics specifically. So one thing I did want to highlight is that in the prioritization process, we asked folks not only to vote on our short list that was under consideration, but also in comparison to the top priorities from 2019. And so 2 of those 2019 priorities are on this list and essentially were reiterated to us as still top priorities. And the other top priorities from 2019 received fewer votes and were sort of demoted. So the feedback that we got was really informative in showing sort of where enough research progress had been made, such that older questions maybe weren't as relevant anymore, and where there was still need, and as well as obviously the prioritization of some of these new questions that had not been included previously. So for 6PBDQ specifically, this is again a figure depicting the number of votes that each question received and the top 5 priorities that we identified through this process. So these priorities are to investigate sublethal impacts of tire-related chemicals, identify additional product sources and the toxicity and degradation products of those product sources, identify safe alternatives and the toxicity and transformation products of those, investigate toxicity of 6PBDQ in various species, including humans, and then what is the effectiveness of stormwater measures for reducing 6PBDQ loadings. So I did want to note that we ended up producing our 6PBDQ shortlist by adapting research needs that had been curated in the recent Interstate Technology Regulatory Council's 6PBDQ essentially synthesis state of the science document. So that was a pretty well vetted list of research questions. And so we built upon that by asking folks to essentially prioritize among those questions in slightly edited form. And so this is the result of that. And the other thing I wanted to mention about 6PBDQ is that we know that there are lots of organizations actively working not just to research 6PBDQ, but also to try to coordinate regional and even national activities in this space and to really keep track of what do we need to know and who is doing work to address these questions. And so we're actively working on trying to coordinate specifically with Ecology and others to contribute our process to the work that they're doing, such as with the 6PBD action plan, and make sure that we are, providing a value add and that collectively we can all collaborate as a region to really facilitate more research in this area and to keep track of the things that we're learning since it's such an active space right now. For microplastics, we didn't actually ask folks in the workshop to vote on these questions. Instead, we met separately with a group of microplastics experts last August and solicited ideas and suggestions from them about what priorities for specifically Puget Sound microplastics research should be. And through that meeting, they helped us produce this top 5 priority list. And the priorities that we identified through this process are: what is the relative distribution of microplastics sources in Puget Sound and the primary pathways? What is the distribution of microplastics among different matrices in Puget Sound? A recommendation to develop a standard method for microplastics, including a focus on tire wear particles. And I just want to note that that tire wear particle recommendation was made because it was noted that the California method development process that's happening at the state level there specifically does not include tire wear particles. So if Washington were to do so, that would be a value add building on what California is doing currently. And then the other 2 priorities are: what is the toxicology of microplastic fibers and how effective are stormwater BMPs at removing microplastics? So overall, what we really heard from microplastics experts was that, there's still a lot of really baseline foundational research that's needed to understand the extent of environmental concentrations and exposure levels in the environment in Puget Sound. So I've highlighted the top priorities for each of our lists, but I will note that our research agenda includes a longer list of questions that are top, high, medium, or low priority level based on the feedback that we got through our expert elicitation processes. And all that full research agenda is now available both in our Grand Uncertainties Matrix database, as well as in a report that we've produced about our process. So I wanted to highlight some of the next steps that we're planning on doing with these research needs now that we've identified them. Obviously, we are really interested in making sure that this research agenda is something that can facilitate and coordinate regional collaboration and energy and funding towards research to address these questions. So we plan on working to communicate research priorities both to the Stormwater Strategic Initiative, obviously, and also to the broader research community of Puget Sound. So our work to provide technical support to the SIL is really intended that the top priorities we identify can inform future funding activities of the Stormwater SIL. So Dustin just outlined all of the great work that they've been able to fund over the last number of years. And so in any future investments that they're able to make, we hope that these priorities can kind of indicate where research might best be directed. And we plan on working with them to really do some sort of cross-referencing of what they funded already, what our priorities are telling us, and where there might be really great opportunities for future research to be funded by them. And then, of course, we hope that broader and additional funding and research pathways in the region are informed by our priorities as well. So, as I mentioned, the Toxics Research Agenda is now in the Grand Uncertainties Matrix publicly for folks to use in making their own funding and research decisions. We have a report that's available if people are interested in reading more about what we did. We are actively presenting on these research priorities to a number of groups, and we've already given talks to a number of different Puget Sound relevant groups like the PSEMP Toxics Work Group, the Science Panel, the Northwest Indian Fisheries Commission's Coordinated Tribal Water Quality Program, and would love to present to any other groups that might be interested in hearing about our work. And then we're currently working on developing a fact sheet about the research priorities that would be a more digestible communication document that we can circulate to interested audiences. In addition to communicating what these priorities are so that we can kind of facilitate some regional interest in taking up more study of these topics, we plan to cross-reference the research agenda that we produced with a recent Puget Sound ecosystem monitoring program, monitoring, inventory, and gaps analysis. That will really help us identify what progress is being made, what existing monitoring might be addressing some of our research needs, and whether there are any that we missed and should potentially add to our list. And then we hope to play some role also in trying to actually address some of these questions. We want this work to be collaborative. We want any number of organizations in the region to study the topics that we've identified as priorities. And so we aim to try to facilitate that effort, scope our own research projects, really assess kind of what level of effort is needed for individual questions and whether convening targeted workshops, doing literature reviews, or doing primary studies would be the best kind of approach for each individual question on our list. And just before I wrap up, I also wanted to obviously highlight the extent to which human health is captured in our research agenda currently. So we feel that our research agenda through our expert elicitation process is very robust with respect to the particular contaminants that are captured in the Toxics and Aquatic Life Implementation Strategy and the fish and invertebrate species that are really the focus of that strategy. But we do acknowledge that our research agenda is not comprehensive when it comes to human health, and that was intentional, and I'll sort of explain that a little bit. About 10 uncertainties out of the 108 on our updated research agenda do include some human health language specifically in the uncertainty itself, recognizing that we, of course, imagine that most of the toxics questions on our list would have kind of downstream human health implications, specifically through consumption of contaminated organisms, but 10 call out human health specifically in them. That includes one top 6 PBDQ priority, which I had mentioned already, which was that question about investigating toxicity in a whole host of species, including humans. We heard a lot of interest in human health topics at our September workshop. People were interested in adding questions about human biomarkers, cumulative impacts on humans, and impacts from other exposure pathways, questions about epidemiology and microplastics. But we consciously didn't add that new content to our research agenda because our scope at the time was really focused on what would be most significant and prove to be the biggest barriers within the implementation strategy. Currently, the strategy only addresses human health sort of indirectly through the consumption of aquatic life. And in the words of the SIL from the last December meeting, human health is not currently a specific endpoint or outcome for the implementation strategy. And that, of course, underpins the work that the SIL is now doing to try to improve the way that we are integrating those things. So based on our remit to really focus on the implementation strategy as it currently stands, we included some human health topics that sort of organically made it into our process through the source material, but we didn't actively include some of the recommendations that we heard about human health. This now at this point has really prompted us to consider, do we need to develop a human health research agenda? Would that be a useful part of the process to develop a human health implementation strategy, which Dustin mentioned earlier? So if any folks have thoughts or ideas about whether that would be valuable for the region to really garner interest in regional focus on human health research topics, that's something that we would be really interested in discussing and learning more about, so that could be a potential project for us to work on in the future. So I'll wrap there. Happy to take any questions or any thoughts or suggestions about human health research agenda work in the future. Thanks.
Thank you, Sandra. That was really helpful. Uncertainties used against scientists all the time. Does anyone have questions for Sandra? Please raise your hand and please speak up. Bob Black asked what the Washington State Department of Health thinks about your work here. We have some folks here. I'm going to call Eleanor. Are you the last DOH person standing here, or Lauren?
I would actually like to call Emerson if he's still in the room.
Oh, is he still here? I'm sorry, Emerson. Here?
And Audrey Coyne from DOH were involved in some of these conversations, and we'll probably have more perspective on kind of this integration and overlap at this time than I do. However, we are having a side chat about how interesting all this is. So Emerson?
Emerson's on. Sorry, Emerson.
No, it's all right. Yeah, so I think, there's been a lot of involvement, but I think a lot of involvement at sort of our level, the toxicologists, and so I guess we'll need to circle back, and if this is going to develop into, I guess, more direct asks of Department of Health and like work products produced, then that's going to have to definitely cycle through management, I suppose, is what I'm saying. And so right now we're in the process of determining what we might be able to contribute and what the new vital sign is gonna look like. The more we can know, and then the more we can get the higher levels communicating, I think the smoother this will all go downstream. That's kind of my my thoughts on that.
Yeah, I think we see a lot of interest overlap. There might on these human health research priorities. We don't, as a toxicology section, we don't have research funds to kind of direct. So to the extent that we can be involved in conversations about research priorities, I think those of us at kind of at our level are interested. I also think, as Emerson says, that as we start talking broadly as a community about greater integration of human health into some of this aquatic life work, that's where it's going to be really helpful to have a little more of a management to management conversation that kind of helps our managers pull together pieces, because we've got shellfish people, we've got us in toxicology work on chemicals, there's drinking water folks, and if we're all kind of working individually on some of these projects, we sort of need to have a little coordination from a higher level in the agency. And I feel like that's not quite there yet. So that's a piece for us to discuss going forward, you know, how to really, where we should be involved in some of this human health integration, how that should look, who the players are, that kind of thing.
And that goes back, thank you, Eleanor, to the earlier conversation about integration, right? Breaking down the silos, and that's like a systematic and also gooey process at some level. Audrey, did you want to add anything before we move on?
Nothing different that's already been said. I think just, yes, DOH houses leads one of the strategic initiatives, but our initiative is very focused on a certain priority around shellfish harvest, shellfish safety, fecal pollution reduction. And so we do find ourselves within the agency trying to navigate and support other health related priorities, but more as like a liaison and networking. But yeah, here to support as sort of these human health priorities kind of seem to be rising as they move forward. But
yeah, thank you. Yeah, this is going to be obviously a perennially evolving conversation. Well, thank you very much, Sandra. We really appreciate your presentation and your work in this area, and we look forward to follow up. So thank you very much. I now want to introduce Hannah Mittelstaedt, who is the network coordinator for the Puget Sound Ecosystem Monitoring Program. The network doesn't work without Hannah. So Hannah, thank you. She's going to talk about the new human health contract and what's going on there. So welcome.
Thanks, Heidi. That was a really nice introduction. Yeah, I'll go ahead and start sharing my screen and get everything pulled up. All right, are you all seeing my
Yes, it
great.
looks perfect. Thank you so much.
Great. Yeah, well, great to be here. Happy to be part of this workshop. So I'm Hannah Mittelstaedt. I'm the coordinator for the Puget Sound Ecosystem Monitoring Program, or PSEMP, as Heidi said. And today I'll be sharing with you about PSEMP's Human Health Monitoring Project, which broadly, this is a scoping project to understand current human health work as it relates to the environment, so we can determine how PSEMP can continue to dive into this work in a really complementary manner. And this is a project that we are right in the middle of, so this will really just be a preview of what's to come. And, you know, I'm presenting on behalf of the project team at the partnership and our contractors at Ross Strategic, but I'm by no means an expert, but excited to be providing an overview. All right, so I'll start with some brief background on PSEMP and then share an overview of the project and its objectives, and then finally the progress so far and what's to come. So in case not everyone's familiar, I'll share a bit about Puget Sound Partnership and PSEMP just to give context for this effort. So the partnership is a Washington state agency that coordinates Puget Sound ecosystem recovery. And part of the state's goals for vibrant, enduring natural systems and communities include ecosystem support for a healthy human population and quality of life. So guiding this work is the partnership's action agenda, pictured here, which is the community's shared plan for advancing protection and restoration efforts across the region. And the partnership also reports on vital sign indicators, which have been mentioned in some of these presentations. And these are a set of long-term measures to communicate about ecosystem conditions. And the vital signs include indicators on human well-being, and the partnership is beginning an effort to review and revise its indicators of human well-being, which is a separate project, but our findings will inform it. And PSEMP is a program within the partnership that supports recovery work with monitoring information. So PSEMP is a collaborative network of subject matter experts who generate, synthesize, and communicate scientific information to track ecosystem conditions with the goal of directly addressing management and science questions critical to Puget Sound recovery. So our strategic plan is a great place to go to find out more about PSEMP's mission, which you can see here. And our objectives are to, 1, increase collaboration across monitoring programs, 2, support adaptive management of recovery efforts, and 3, improve communication within and beyond monitoring and the monitoring and assessment community to improve access to credible information and guide recovery decisions. So the PSEMP network is made up of topic-based technical work groups. You've heard the toxics work group mentioned. And the work groups are the primary venue to assemble the many entities from across Puget Sound that are responsible for and implement environmental monitoring. So each group is focused on a key monitoring topic in the diagram here, and these groups provide spaces for subject matter experts to collaborate to determine monitoring trends, make connections across disciplines and data sources, and identify monitoring priorities and gaps, and much more. So what Sandra presented is a good example of some of our involvement in that. So PSEMP is supported by partnership staff, including Katrina Raddick, the PSEMP manager, and me. And we also have a steering committee that helps guide and elevate our work. So some members are here today, including Heidi. And PSEMP has historically centered around monitoring of species, habitat, and water quality conditions, and not specifically on human health or human quality of life. So this project aims to help PSEMP enhance existing human health monitoring efforts and identify opportunities for new monitoring of human health exposures and outcomes. So to dig into the objectives of this project, the overarching goal is to better understand the current landscape of human well-being monitoring efforts, including those related to human health, by identifying existing work, available data, and critical information gaps. So this is in addition to the current vital sign indicators associated with human health, which are shown here. And to do this, we are partnering with Ross Strategic. They are contracted to carry out this project, which started in January and will wrap up in September. And the project is led by Jen Major, and other team members include Elizabeth McManus, Peter Rabinowitz, Michaela Unda, and Summer Khan. So we're very excited to be working with them. And the project will explore the feasibility of monitoring indicators of human health outcomes from toxic and biological contaminants in Puget Sound's air, water, and food, extreme temperature and other human health threats posed by climate change, and other relevant Puget Sound environmental exposures or impacts. And products from this work will include an inventory of current data and efforts in the region, insights on current gaps, and recommendations to the partnership and PSEMP. So this is a preview of some of the data categories and a few of the sources the Ross team has already identified for its draft inventory. There's a lot to consider, and this slide is just scratching the surface, so a lot more to come. And to give you an idea of where the team is and where they're going, this is just a high-level overview of the timeline for developing the inventory. So far, the Ross team has conducted background research and begun developing their outreach plan and gathering information. They are currently reaching out to schedule interviews and focus groups with experts in climate and health, toxics, One Health, zoonotic diseases, environmental health, and other related topics. All to better understand existing data sources, current monitoring gaps, and opportunities for tracking human health outcomes related to environmental conditions. So they met with the PSEMP work group leads last month, and will meet with the PSEMP steering committee later this summer to share updates on themes and findings. And after they conclude outreach activities, they'll develop an outreach process summary, and further develop and refine the inventory. So in September, the inventory and recommendations will be finalized and shared. So to summarize the Ross team's progress so far, they have developed a draft inventory of existing data sets and disseminated an online form to gather additional data for the inventory. They also worked with partnership staff to refine a list of contacts for interviews and focus groups. And in the coming months, while they conduct interviews and focus groups, they'll engage with the PSEMP work groups to get their input on the inventory too. So this is an ongoing project, so expect more to come in the future. And ultimately, we hope that this project will provide insights that will help the partnership align its investments and partnerships more effectively and support priority actions that foster a thriving ecosystem and healthy, resilient communities. So thank you so much. And please reach out to Katrina or myself with any questions or feedback. And with updates and for updates on the project and all things PSEMP, please sign up for the newsletter, which I'll put a link to in the chat.
Thank you. Thank you, Hannah. Does anyone have questions for Hannah about this work? It's really great and it's very exciting. And I just want to say that this conversation, oh, I see, Eleanor, that you're on, has been going on for a long time. So it's been sort of an accreted evolution and very positive. It can be difficult to move from a certain domain where you've invested a lot of resources and then to start coloring outside of the box to expand that, and change is never comfortable. So we're really grateful for the willingness to take some risks here. So thank you.
Thanks,
Eleanor, do you want to ask a question?
Yeah, I do. And you know, understand, like, I haven't been involved in this work, so I'm kind of coming at it a little peripherally here. You know, my colleague Emerson Christie has probably had some input to some of these conversations, but I guess I'm feeling like one of those I saw, and there's like biomonitoring, and I don't know where that data stream is, but I feel like that was a DOH website. I mean, we had a one-off biomonitoring grant many years ago. We would let me just say, we would love to have a biomonitoring program in Washington like there is in California. I mean, that would be fantastic. But that's not something there's budget for or legislative will for. So I don't see how that could be used as a time trend piece of information. So couched in that comment is kind of the question, you know, where are the connections to DOH and some of those data sets? I saw a lot of Washington Tracking Network, like are our tracking network leads like in the conversation here? I think it goes back to what Emerson and I were just saying. We wonder if our connection structure into this work is maybe not happening at the right level at this time. Maybe we need to work together on kind of figuring out those intersections and kind of who needs to be at the table and where those conversations need to take place, like keep these intersections going. Because yeah, just like the biomonitoring thing was really like, whoa, wait, what? For me right in that moment.
Yeah, thanks. That's all super helpful and yeah, great feedback and great questions. And I think you know, for this effort, the team really is taking like the widest lens possible. So they're just trying to see, you know, what has been done in the past, what is continuing to be ongoing. So that the scope is quite large, and then we'll you know, narrow down as we identify, you know, what current monitoring efforts are and where where energy is like currently being invested in everything. So those are my thoughts for your first question. And then for the second part, I know I was looking at the team's contact list for subject matter experts for the upcoming focus groups, and I did see DOH folks on there. And yeah, happy to talk to you or get you in contact with the team to make sure that the right people are involved in this conversation. And at the right levels, because yeah, this is, you know, we're in the early stages of the project and everything. So yeah, happy to have that conversation sooner rather than later.
Okay. Yeah, that's great. Thank you for
connect
that.
you.
Yeah, I think, you know, like Emerson and I aren't on the Washington Tracking Network team. That's a whole separate body of expertise. Like there are bins of expertise here that I feel like you're starting to touch on, and I just want to make sure that we're in the conversation the right way. Emerson, did you have something to add?
Oh,
Yeah,
and then
I was just
Marguerite, and then we have to move on. Sorry. Thank you.
yeah, I was just gonna say, Hannah, like, we can, I've already been on one of the meetings.
Oh, great.
And so, like, and this is kind of, where I'm starting to pull in other people to figure out the placement here. But part of it does depend on like work product and work outcome and what, one ends up. If there are going to be asks of DOH, then it really needs to probably start with a management to management connection. And then, for the but for providing feedback and input, we can still do that. But I think it might even be more effective even with a management to management connection, because then you'll really get down to the correct people for the correct meetings. So yeah, anyways.
Yeah, great. That's super helpful. I'll be sure to pass that on.
Yeah.
Thanks. Marguerite?
Thank you, Marguerite, and then we'll move on to Dustin before we
Sure.
close.
Yeah, no, I just want to say the group that's been working on this has been doing a great job. And they're into weeks into really trying to reach out and get advice and input on who they need to be involving, what levels, names, places in the organization, both at the state and county levels, because some of the data that may be relevant might be more to the point if it's when collected at the county than even at the state. So Eleanor and Emerson and others on this call who have, who now are starting to see the bigger picture and have real suggestions for who Jen Major and her group, Peter, who they ought to be reaching out to via, Hannah and Katrina. Please send that input in. They've tried individual emails, they've tried going to conferences, they've put up a form for people to fill out and send in. And so they are trying their best. So we need everybody's input on this. So happy this has caught your attention. And yeah, they have open ears. They are ready to hear it so that they really can deliver the best product on this. So thank you, everybody.
Yeah, thank you. And for
Yeah.
those of you who don't know Marguerite was like a big kahuna at CDC epidemiology. Still has a foundation, I think, with great photography associated with the foundation page. But yeah, thank you, Marguerite, because you've been like a total radical mover of all things health here. So it's been amazing. And thank you, Hannah.
Yeah. Thanks so much. Yeah, this definitely feels well timed with where the project's at right now, where it is, if you're interested in getting in contact, if you know someone that you think would have good input on this, like, yes, please reach out, because that is a major component of where we're at right now. So yeah, thanks so much for that, Marguerite, and thanks, Heidi.
Thank you. We're going to move on now to Dustin and his poll. So let's take the poll and we'll wrap up. Thank you for hanging in here.
Yeah, thanks, Heidi. And this will be just real quick. Want to remind folks that if you have time and want to provide feedback on the draft vision statement that I shared at the beginning, the link is in the meeting packet, and I would love to have your feedback by the end of this month. We're going to be following up soon with notes from today, links and the recordings and everything, so be on the lookout for that, hopefully before the end of June. And we're looking at a time frame of like September, October for our next meeting. So I put a link in the chat where you can add your thoughts on what you want to hear about in upcoming meetings. There's a lot of things kind of like going on in this great swirl of human health activities in Puget Sound. The Puget Sound Action Agenda is continuing to move forward. The Shellfish and Stormwater Strategic Initiatives led some of those topics in the conversations, and those will be carried forward, and you'll be looking for those in the action agenda when the partnership has the updated draft for public comment this fall or next winter, I believe. And then also going to be, and so we're kind of like seeing how those things shake out before we start getting too deep into updating the toxics and aquatic life strategies. But that is something that I want to be doing here later this year and next as well.